Did you watch it?
All right, I'm just recording from the get-go.
Did you watch the supplementary movies?
I did, and they're good, and they'll make a nice little... I feel like I never really see the supplemental movies until I'm editing together the video for the YouTube upload.
Oh, yeah.
And then I really watch them several times because I'm uploading them. I'm going to just read out loud this mission statement that you wrote up. I've been thinking about this paper a lot over the last few days, that I'm quite worried. This is the second coolest paper that we've covered. I'm just going to read the mission statement and then we can start the episode.
Okay, sounds good.
Mission statement for the Lemma Journal Club. The primary goal is for Bezia and Linnea to spend time together talking about shared interests. We record it to give to our daughter, Asia, someday in the hopes it gives her some insight into who we were when she was young. So the primary goal is to spend time talking together about shared interests. We are also aware that people in the science community will listen and react. We want to be forces for positivity in this aspect, while still being able to have meaningful criticisms. We should be both unafraid and enthusiastic in our criticisms. Our friends and family are another audience we know is listening. For this, we want to be entertaining and give them a sense of what we spend our days thinking about. And finally, young graduate students and budding scientists may also listen. It is for them, as well as for us, that we should remain candid and unafraid in our criticisms. Okay.
Should we intro?
Yeah.
Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number
11
of the Lemma Journal Club.
What is the Lemma Journal Club?
No, I think that blew out the microphone.
Well, we're going to just find out. We can just cut that bit.
This is not a good intro. The Lemma Journal Club is the show where we read every paper to tell you if you need to read it. You probably do.
We have a terrific paper for you today. We are talking about the research paper, Cytoplasmic Division Cycles Without the Nucleus and Mitotic CDK Cyclin Complexes. It's by Anand Bakshi, Fabio Echegaray, Andrew Alamban, Miquel Rosas-Salvans. Miquel, I can't tell from the font if that's a G or a Q, I'm sorry. Sophie Dumont, who has showed up on quite a few papers that we've covered now, and Mustafa Aydogan. Linnea, why did you pick this paper?
Yeah, so I picked this paper because the first thing I did when I joined a biology lab for my postdoc, or the first experiment that I really felt was successful and beautiful, was watching Chlamydomonas divide. And I had the purinoid, this chloroplast localized organelle, labeled, and I got to watch it divide into its daughter cells. And it's kind of a mesmerizingly beautiful process and brings up a lot of questions about how it's all coordinated by the cell. So a Chlamydomonas has a single chloroplast and also a single nucleus and a single purinoid. And each daughter cell has to end up with a single nucleus, a single chloroplast, and a single purinoid. And so how does the cell coordinate all of these divisions at the same time? And yeah, I saw Mustafa give a talk at an HHMI conference about this work, which is looking at the timing of cell divisions in a multicellular organism, Drosophila. And yeah, it reminded me of this initial beautiful experiment that I did and the curiosity I have about how the cell coordinates division timings.
So a quick question. This paper is about cell divisions that happen where there's no nucleus in the cell. So you just have two fake empty cells that are, you have a fake empty cell that divides into two fake empty cells.
Yeah, so I didn't know that was possible.
But my question for you is, can this happen in Chlamydomonas? Can you get a zombie Chlamydomonas, which has no nuclei? And then if you have a zombie Chlamydomonas, can that Chlamydomonas divide? Yeah.
Yeah, I have no idea. And then, one of the really interesting things to me is that, okay, plants have multiple chloroplasts per cell, and those chloroplasts divide on their own timing, right? And also, you study mitochondria, and this is, again, a place where we're trying to find, or a time where we're trying to find overlapping interests. And mitochondria also have to divide, and they seem to do that with some timing that's separate from nuclear division. And so I was excited by the movies that Mustafa showed during his talk of the cytoplasm dividing independently. And I wanted to read up on the details. So I used this journal club to force me to read this paper.
So this is a terrific paper. I said that at the beginning. I say it about each paper. Every paper that we cover is a terrific paper. Otherwise, we get about a paragraph into it. I say, we're not going to do this paper, and we go and find another paper. I had tremendous difficulty reading this paper, and I'm going to talk about that a lot. This is probably the most interesting paper we've done since mitochondrial purling. The more interesting the paper, the more critical we're going to be of the paper. So if we listed your name... I don't know if
we should say we. I don't know how critical I'm going to be of this paper. Okay. I'm mostly enthusiastic about it. Okay,
then you're going to be good cop. I'm going to be bad cop. As bad cop, if I listed your name during the authors section, please turn off this podcast. I mean, you can keep listening. I think people should be aware of the way that I speak when I'm excited about something and how self-critical I am. We're just still getting blowback from the mitochondrial purling episode.
Which we love that paper. It was such a beautiful paper. We think about it all the time.
I think it's just a difference in culture. Maybe it's a difference in like biophysics versus normal biology. So anyways, we reread our affirmations of like what we're trying to make this podcast at the very beginning. The other thing that I want to say before we totally dive into it is that there exists a podcast on this paper already.
Oh, yeah.
There's a podcast series called Preprints In Motion, which is doing interviews with authors while the paper is in a preprint stage. So this was recorded about 3 years ago while this paper was still a preprint. And there's interviews with Anand, Fabio, and Mustafa, I think. I'm just doing this off memory. They have 100 episodes. They have a lot of episodes, Preprints In Motion. They've been going for a couple of years now. So if you're the kind of person who's listening to science podcasts, you might consider checking them out. Do you know, by the way, Sophie Dumont?
No, I was just looking at her affiliations because you mentioned that she had been on other papers as well.
She's UCSF.
Yeah, and Chan Zuckerberg. I have no idea. I hadn't, the name looks familiar, but
she's also, for instance, the reason why I said she might be sick of us is she's also on the mitochondrial purling paper. Mm-mm. It's someone who's obviously, I think, close to us in our field.
And I guess doing exciting work.
And doing very exciting work.
But I don't think I've ever met her. I was super lucky to have met Mustafa even. So.
Where do we want to
start
on this? I guess I'm going to start with what I highlighted in the abstract before I had read the paper, which is that this is a paper that I would say is very up my alley. My PhD lab studied the spindle during cell division very tightly. And then my postdoc lab, well, I don't study this, studies multinucleation events in cells being cultured to kind of look into multinucleation during cancer.
Where multinucleation is multiple nuclei?
Yeah, so the nuclei divides.
Mm-hmm. But the cell does not.
But the cell does not.
Yeah, okay.
And so you get multiple nuclei inside a single cell.
So it's the inverse decoupling.
And so this is Miriam's big project. And just from the very beginning, even though I have that background, I immediately underlined in red cytoplasmic divisions and nuclear divisions and wrote down to myself, wait, what? Uh-oh. Because I don't normally think of divisions as nuclear divisions and cytoplasmic divisions. And so I'm in the right labs to think about this. But already I'm a little bit caught off guard.
Wait, what do you mean? What do you think of them as?
Well, they say the cytoplasm starts dividing. And I was like, what do you mean the cytoplasm starts dividing? It's just like a nomenclature that I've never had in my head before. Of like, I always think the cell divides.
Yeah.
Or the cell fails to divide. I don't break it down into the nucleus divides and the cytoplasm divides as two separate events. And so there's already a big restructuring in my mind just from the abstract, just to get through the words that the abstract is saying.
My nitpick from the abstract is something that came from you a long time ago. So we wrote a lot of papers in our PhD about microtubules. And for a long time, like in our field, we would abbreviate them MTs. But we had a conversation early on in grad school where you were like, this is unnecessary jargon. Just write out microtubules every time when you're talking to a broad audience. And that really stuck with me of just like, don't put in extra acronyms when you don't need them. Microtubules is not a long word. It's not increasing your word count at all. It's still one word. And so the use of MT is just like a pet peeve for me now.
The reason why MT came up in our first or second year of grad school is I started working in a clean room. And in the clean room, when you have an empty bottle that's ready for disposal, because disposal is a whole rigmarole in a real clean room, you label it MT. just like you would. And so I came in with a bunch of active matter components and some microtubules labeled MT, and people threw them out. And it took me a while to understand what was going on. And then I understood what was going on. And I was like, oh, this is the classic scientists have named something twice.
Yeah.
And I'm the whole clean room, which is many, many different scientific fields, has agreed on the letters MT stand for this is empty, please throw it out. Only my extremely narrow subfield thinks that MT stands for microtubules. So I'm just going to do it their way.
Yeah, how do we want to get into this paper? It's such a large paper that I guess the thing I had wanted to say, and I had texted to you earlier and you told me not to say it yet, is that reading through this paper, it felt like a paper that one of my bosses could have written. Just in that, yeah, the way it was presented of like all of this evidence for this statement, all of this evidence for this statement was a very like classic biology, classic cell.
So I think we start on page quote unquote 3, right before cytoplasmic division cycles can run without nuclei independently of the mitotic CDK cyclin complexes, because I have a comment that says actual paper begins here, that I feel that the first chunk of results is just rehash, where basically they're saying these results suggested that cytoplasmic divisions could occur without a nucleus in fly embryos, which they can do in 1 millisecond in a video where you see a cell which has no nucleus and you see that cell divide. And what we really get into is how does it divide? What's dividing it?
Well, so you say that this is where the paper starts. Is it not where the paper ends?
Yeah, that's the other way to view it. The reason why I say that this is where the paper starts rather than with this is where the paper ends is to some extent, it was already known that cells could do this, right? This had already been done. I don't know how much I really want to say that. It's again like the mitochondrial purling thing.
I was not aware of it before this work.
Right,
it doesn't mean that it wasn't known by people, but I was not aware of this process, like
Right.
of a cell divide, of a cytoplasm dividing without a nucleus before this.
Right. So I think similar to the mitochondrial purling paper, our takeaway from that paper was really loved the framework that they developed. But they were not the people who discovered that mitochondria were purling, nor were they the first people to invoke this Rayleigh plateau instability. In a similar manner, they talk about this both in the introduction and in the conclusion and on the podcast. There are studies in zebrafish, in canine epithelium. I think maybe even other studies in Drosophila, although I might be wrong about that. From the podcast, it kind of sounded like this was a thing that they saw in Drosophila that other people hadn't seen before, where you had cells without nuclei dividing. So that had been observed before. And I would say that rather than the big finding here being that this also occurs in Drosophila, I would say that the big finding here is learning doesn't need CDK1 cycling.
Yeah.
Does it need microtubules? Does it need a centriole? Does it need a centrosome? Does it need myosin? Right? And like, what are the things that are needed and what are the things that are not needed?
Yeah. What is doing the coupling, I think, is kind of the driving question of the paper.
Yeah. And so that's why I say paper starts here is when they start messing with CDK1 activity.
Yeah.
Yeah. And it's just like, I think from historically that the basic phenomena was already known.
Okay.
But, so yeah, the first thing that they do is they have some ways that they can change CDK1 activity. Always with flies, I'm not sure, CDC25 and CHK1, like to what extent are these things known that they work? But, they mess with CDK1, they get comparable cytoplasmic division dynamics. So they say it's uncoupled from CDK1. I found this not super convincing. They go back and do it a couple ways. One way that they go back and do it is with a CDK1 specific sensor.
This FRET sensor.
Yeah.
To show that it's active or it's inactive.
Yeah.
I think that must have been, maybe it was a control that they did internally of like, these results don't really make sense. Let's make sure that our CDK is actually being inhibited.
Okay. So they then proceed to say a bunch of statements that I had difficulty understanding. And so one sentence that I did not figure out what was going on, they said, like in a regular blastoderm, cytoplasmic compartments in arrested embryos also became ensheathed with plasma membrane. What does that mean?
Like in a regular blastoderm, cytoplasmic compartments in arrested embryos also became ensheathed with plasma membrane. Figure 3A, middle row. We're on figure 3.
Yeah, figure 3A. Ensheathed in plasma membrane. Isn't the plasma membrane just the... I guess cytoplasm is the inside and plasma membrane is the surrounding side. But what are we... I mean, the plasma membrane is the compartment. This seems tautological. That, like, if you're not ensheathed in a plasma membrane, you're not really a cell. But I guess that's wrong. Like, Drosophila particularly don't always have plasma membrane on all four sides of their... or all sides of their cell.
Yeah, I kind of glossed over this.
Yeah. Okay. So now you glossed over this. Let me make my chief complaint about this paper. There is no way to look at these figures. They're unparsable. I read the paper. I looked at the movies. I read the figure captions. I really tried to look at the figures. I tried until I had an actual legitimate headache. But they've got fonts describing what each figure is, such as myosin regulatory light chain GFP, in size 5 font. The figure is enormous. It's the whole page. And so I cannot, and the text information is coming from several pages before, or because it's cell, they do this incredibly obnoxious thing where they take the figure caption and they split it in two. So it becomes this game of like, where's Waldo? Or like a magic eye or something where I have to remember what I'm looking for, go to the figure, zoom way in to find the part of the figure that I'm going to read, move around it like I'm a cartographer, go down to the figure caption to figure out what's going on, go back up, and I really was unable to keep all the necessary pieces of information in my mind at once to parse the figure. And I blame not the authors, I blame the editor or the person who's doing the printing. You need to be a falcon in order to read this paper appropriately. And because the figures are so huge and the font is so small, I feel like I'm studying a map of the world with a magnifying glass looking for Springfield in America, right? It's just like getting the context is so hard. I think my first comment is, oh my God, why? And my second comment was, I think this was the next day, I said, if it wasn't for Journal Club, I would close this paper at this point. This is a great paper. This is cool science. But why would the editors allow this type of figure?
Read other cell papers?
Right. So this is why I say this is the editor's fault. This is the publisher's fault. This is the proof's fault, is that Cell keeps doing this.
I think this is just what a cell paper is. Like, a cell paper is not something that's easily digestible.
But it's not just not easily digestible. I would argue that someone who needs reading glasses, a 40-year-old, cannot it is physiologically impossible for them to digest a cell paper. So that nobody who's over the age of 40 has ever actually read a modern cell paper. That's my claim. 45-year-olds come out and disprove me, but people, it's just so hard to get information from these figures. And then trying to figure out what arrows meant. I just went to the movies. I read the captions of the figures. I got super frustrated from this experience.
The way that I dealt with this, because I the figures are huge and there's a lot of really subtle things happening in the paper, which I think to look at the images and see what they're seeing, takes a lot of looking. So it's not easily digestible data either. The way that I read this paper was I read it front to back text only.
Yep.
And then I went through on the web page and scrolled through the figures, and they put the supplementary figures, the supplementary movies, and the main figures in line. And so I scrolled through them that way and played all the movies, which I think is a way easier way to digest this data.
Yep.
And then I went back to the figures and was like, okay, let's see if I can now parse what's going on here. Especially so figure 1B, they have a beautiful movie, but I did have trouble in the movie seeing this division, this cytoplasmic division furrow that they highlighted. And so I went back to, that's where I really got into the figures, was in figure 1B. I was like, okay, so I saw that you had this movie, what are you actually pointing at? What's actually happening? And so then I looked at this figure I think it's a large figure. It could have been edited down to be more easily read, but I was not surprised by this in a Cell paper.
I wasn't surprised. I would say almost always for any other journal club, if someone gives me a Cell paper to read, I read through the text, I look at the movies, and I usually give up on the figures. And because we're recording this, I really sat there for hours trying to decode the figures. I did it almost essentially the same way that you said you did it. I read through all the text and all the figure captions. Then I watched the videos, and armed with that information, I started to try to capture the actual figures. But you imagine, right, from a writing perspective, normally the first thing that you make is the figures. And then you write the text to follow. So I'm like almost reading it backwards of how they probably wrote it. And I think that this is a fundamental problem with Cell. So in a normal journal club, I would not look at the figures, and I'd wait for other people to point out cool things about the figures. And because we're recording this, I was like, I've got to go the extra mile. Normally when we do these journal clubs, it takes me about the normal amount of time to read a paper as I'm reading every week anyways. This was like a real time sink this week to try to get through this paper. They did for the videos, which are very nice videos. They don't have annotations on the videos, so you do have to go to the figure to look at their arrows and to see what they're pointing at. Because the videos are very hard to capture the cytoplasmic things. I also got concerned in many of their figures going down in Z-depth, they have shockingly low Z-depth penetration. Like they're losing a lot of signal 5 microns into these cells.
Well, it's a spinning disk.
It's a spinning disk.
Yeah.
Even for a spinning disk, I mean, that's good to know. Even for a spinning disk, I thought you would get 40, 50 microns into these cells. I guess with Drosophila, I'm always a little bit not clear about the staging, that there's a lot of staging in which the like outer shell of the embryo, of the egg, the outer shell of the Drosophila, whatever it is extremely optically thick and obnoxious. And I encountered that a lot with my own Drosophila imaging. And so I guess I don't know where we are there. But yeah, I'm trying to remember if I ever did spinning disk imaging of the Drosophila. I'm quite surprised that they're losing information so quickly.
I'm not that surprised because we can lose information within a clammy cell.
Okay.
If it's dim, right?
So then my concern then, if you're losing signal that quickly, you're losing it within a single clammy cell is crazy. Yeah, and clearly things are dim here. My concern is that sometimes there might actually be a nucleus or a centriole or something in the cell that they don't capture because it's just lower than they were expecting it to. They have in figure 1b, an XZ slice, and it doesn't really look like they're ever able to capture the cell membranes on the bottom of the cell, which makes me concerned that they could be missing something. To be clear, I'm not actually skeptical of anything in this paper. I normally say like extraordinary claims require extraordinary evidence. I don't think that this is an extraordinary claim. Once you've put this out there and I've heard it, I was like shocked at first, but I think it's quite reasonable. I actually don't think that they ever say anything in this paper that isn't the way I would have guessed it goes. And so I'm never upset that I don't have extraordinary evidence here, but I do worry that we don't have a full 3D image at the right resolution here. People have made the same complaint about my mitochondria data in Drosophila, that I make some claims of that the mitochondrial content is going up, even though it's not supposed to, and that there are more mitochondria in some cells compared to other cells. And people are like, oh, your 3D resolution is not good enough. And I would say my 3D resolution in those videos are better than the 3D resolution here. So the victim of my own complaint, but... Yeah,
I do wonder, there's a ton of light sheet data on developing Drosophila embryos out there. I mean, maybe not with this actin labeled, but with some sort of cell membrane marker. I wonder if you could see this in data that's already 3D resolved data that's already out there.
These markers for Drosophila are very bread and butter markers that I mean,
the wild type behavior, not obviously all the inhibition experiments.
Yeah, I actually have I have videos of ventral furrow and cephalic furrow formation in Drosophila using some of these markers just because they're so bread and butter.
And what did you think about them using myosin as the marker for this cytoplasm division?
Yeah,
I was surprised that they didn't use a membrane marker, which to me would have been the obvious edge of the division, but maybe I'm misunderstanding what division
I think
is
it was a
defined
convenience
as.
thing. It's just that everyone has this MRLC, GFP, Drosophila in their lab.
That's been shown to be correlate. Like naively, when somebody says cytoplasmic division or any cell division, I'm thinking that the compartment divides into two new membrane enclosed compartments.
Yeah.
Does this myosin track with membranes? Is that known? Or is this saying that a furrow comes in and squeezes things off before membranes get...
Yeah. So I guess I'll say from my own myosin imaging, right? Any cell can be partitioned just from the myosin signal, right? Because the cell is surrounded by actin, or the cell-cell junctions, are outlined by some actin, and there's going to be some myosin where there's some actin.
Yeah.
There will be significant myosin enrichment in certain locations where the myosin is really doing something. So for instance, when I see a ventral furrow forming, I will see a lot more myosin on the apical side of those cells where that's driving the actual furrow formation. But I can still draw you where the rest of the cell is using the myosin signal.
Okay.
So I don't think it clearly, I would say, visually works here.
Yeah.
That you're clearly outlining cells successfully here.
And then the one thing I want to say in response to this Z resolution complaint is that all of their experiments have the same problem, right? Where they can't see this bottom membrane.
Yeah.
And they're seeing the same behavior, like all their inhibitions are done like compared to wild type.
Yeah.
So I don't think it actually impacts their findings. It just impacts like whether the initial event was happening.
It creates the point of like comparing the drug versus the non-drug is salient. It just creates a bit of wiggle room. For instance, the drug might create a phenotype in which it moves really low down onto the like basal side of the cell or some garbage. I'm not saying that occurs. I'm just saying I was surprised at how quickly the signal goes away and concerned about how quickly the signal goes away. I wouldn't, as like a reviewer, mention that or anything. It was just a thing that I noticed.
So one thing I found really interesting in this paper was how many negative statements they made. So the first title, or where you say the paper begins, the first title is Cytoplasmic Division Cycles Can Run Without Nuclei Independently of... Can run... Oh, no. Never mind. I would like to cut that. For the second one... the cytoplasmic division cycle can occur without centrosomes in developing embryos. So this is a negative statement, right? Where they're saying like, yeah, this thing is happening without a structure. I feel like that's a hard thing to prove.
I don't know if that's a hard thing to prove. Although it does. Okay, so it brings us to how do they prove a lot of this like centriole centrosome stuff? The way they prove this stuff is they ablate the centriole. And the way they do that, you said this was all on a spinning disc. So did you read what the actual ablation process was?
I didn't. I meant to, but I didn't get to it.
So hopefully they come in with a little laser and they go, doo-doo. And I'm always a little concerned about how people do ablation, but sometimes they do it right.
They have a lot of controls in their supplementary figures.
I'm just going to assume that they do it right here. My question is, how do they know that they're not just photo bleaching the centriole and then just messing the cell up by hitting it with a lot of heat?
Well, they did a control where they ablate right next to the centriole.
Yeah.
And they say that doesn't impact.
It doesn't do heat. Yeah.
The cell cycle. So that was their control for that. And then I was concerned about verifying that they had actually ablated the centriole.
I mean, I guess there's not a big difference between ablating the centriole and just messing it up. It doesn't really matter if they're messing.
Yeah, but if you're just bleaching it, if you haven't like fully ablated it.
But they see a difference occur.
No, they see no difference occur.
Is that true?
Isn't that's what I'm saying? It's a negative statement. So they still see this division happen even when there's when they've ablated the centriole.
But I thought there was a result here where it's like, you can do the division without a centriole. You can do the division without CDK1. but you need to have at least one of the two or you don't do the division.
Okay.
And so for that experiment, presumably somewhere here in these completely unreadable figures, somewhere here is an experiment that shows CDK1 inhibited, we ablate the centriole, and the cell divides. There's no way to know because there's no way to read these figures. I mean, there is, right? But it's like, where's Waldo? We'd have to sit here for 30 minutes trying to find it.
Yeah, so they do show that when they ablate the centriole in a different fly, that it messes up the spindle formation or what? Yeah, like the organization of the microtubules. So they are, in theory, able to ablate centrioles. It's just about whether they're actually ablating them in the experiment that they show. That was my question or concern.
They have this claim centrosomes are neither necessary nor sufficient to trigger cytoplasmic divisions in developing fly embryos.
Yeah, this experiment confused me. So they took unfertilized fly eggs.
Yeah. I forgot about this. Oh, yeah. Okay, keep going.
And they try, there's a way to induce centrosome formation. So centrosomes are the whole structure, and centrioles are the microtubule.
Centriole are the weird triplet microtubule core structures.
Yeah.
Which they always show an image of two tubes. And I do wonder, is it always literally two tubes? Or is that just like a motif and there's some number of tubes?
So a centrosome will have two centrioles. Yeah.
Well, is it two centrioles or is the two
centrioles.
of them combined called one centriole?
No, two of them combined is a centrosome or part of the centrosome.
Part of the centrosome.
Yeah.
And each individual one is a centriole.
Yeah, that's what AI told me earlier today.
If Dan is listening to this, he must be groaning because it was 6 years of group meetings in which people were describing centrioles and centrosomes. But I was never 100% sure on what exactly the centriole, if it's two, if it's one, if it's the whole structure. But centrosome is the whole thing.
Is the whole thing.
Yeah.
So they take an unfertilized egg and they treat it with something, or they genetically perturb it in some way to create centrosomes. And then they don't see compartmentalization of the cytoplasm. Mm-mm. And to me, this was why would we expect that to happen?
Yeah. So this is, I think, so there's this idea in biology. I'm going to say it's like a philosophical idea about if something is necessary and sufficient. And when this was described to me, I was like, ooh, this is a really nice intellectual tool. Is this necessary and sufficient for something to happen? Right? But when you actually start to apply the words necessary and sufficient, one of two things has to happen. Either you have to get sloppy with the words, in which case the tool is broken, or you have to make super, super narrow statements. So if you have a ball going down a ramp, right? And you say, is the ramp necessary and sufficient for the ball to go on this trajectory down the ramp? And necessary, yes, right? Sufficient, I think a lot of biologists would say yes. But if you want to take it super literally,
and
you
would
be
just move the ball with your hand in the same trajectory.
You could just move the ball with your hand, so it's not necessary, right? And sufficient, you could also be well, you need the ball as well for the ball to move. And you could also say you need gravity, right? And I think the tool necessary and sufficient is a beautiful philosophical tool that is not being used well in biology. And sometimes, actually, in this paper, they use the term necessary and sufficient a couple times in super specific manners where it really does fit well. And then it's a super useful tool. But if you say something like centrosomes are neither necessary nor sufficient to trigger cytoplasmic divisions, right? It couldn't be sufficient because you need the cytoplasm as well, right? The centrosome cannot in and of itself be sufficient to do something that requires the centrosome plus another thing, right? Tautologically, it's not possible. So this has nothing to do with this paper. This is not a criticism of this paper in any way, just for the authors who at this point are fuming at us. This is a criticism of biology on its own term that I loved this concept necessary and sufficient when I learned it in biology. And now that I've been using it for a couple of years, I am less enamored because I feel it is not used in a philosophically consistent way.
Yeah, so I can see the appeal of doing this experiment, right? Because if you were somehow to take an unfertilized egg and be able to induce a bunch of cytoplasmic compartments just by creating centrosomes, that would be amazing and a beautiful result, right? But I think the negative result, which is what they show, is so it's saying that it's not sufficient in the unfertilized egg, but it's not saying anything about the fertilized developing system, which is what you're actually studying. And so that's where I felt like it was, a confusing experiment to me.
There is, to some extent, I'm just looking at I'm a little bit low on all these audio. So maybe next time I have to put my audio up higher. There is, to some extent, they talk about this a little bit in this podcast interview that they do. There's a lot of fundamental questions here about what's going on that I'm going to say, in my opinion, and I think in their opinion as well, are inaccessible in a whole organism.
Yeah.
That you have to go to cell culture to start banging away at some more fundamental things here.
Yeah.
So there is something to be said about like doing experiments that are getting further and further away from the organism that you meant to study, from the natural environment that you meant to study, just because there's so many little things here that are kind of mysterious. While we're on figure 4, I'm just going to one last note. At the bottom of figure 4, there's a little sentence that says, statistical significance was assessed using a Welch's t-test for Gaussian distributed data or a Mann-Whitney test, scale bar 5 microns. This rule that all these journals have recently rolled out, where I have to say what tests I used to calculate the statistical significance in the figure, in every single figure, is a garbage rule. Again, it has nothing to do with it. Like, this is a rule of cells. You have to put this. It's also a rule of science. It's also a rule of nature. You have to put this statement in every single figure rather than in the methods, which is where it belongs. And it's just been making me think about, we talk a lot about how much recently the journals are starting to waste our time of saying your metadata must look like this, and it must be annotated for every image, and you must put this here, and you must put that here. And there are, it basically doubles the length of writing a paper to meet the journal's random criteria. But the other thing that I've realized that I'm starting to bother me is not as a writer, but as a reader, the journals are messing with how I get to read the information, that they are filtering what information I see and how it is presented to me, so that now at the end of every figure caption, I have to read about the Welch's t-test and the Mann-Whitney test for the 8,000th time. And I do not like that the journals are making decisions of what I get to see. I really want the writers of the papers to make the decisions of what I get to see. Because I don't like this thing where I'm now criticizing this paper about a thing that they didn't have a choice about, right?
Yeah, it's just a waste of time for me.
It's not just a waste of time, though. It's making my reading experience worse.
Yeah, I hear you. Yeah.
So
my rant against p-values is like they show the violin plots with all of the data. I don't need a p-value. Do I?
I don't know. A lot of biologists are going to fight you tooth and nail for that. What Eric says.
And then sometimes I get p-values that are like 10 to the negative 30. And the journal comes back to me and says, you need to report the exact number. And I'm like, what do you mean? If it's 10 to the negative 30, that's all I can say, right?
Eric would say, for instance, for figure 4b, that
our eyes can deceive us and we're not.
No, that's not what he would say. He has a very good way to think about this that I like.
Okay.
And so I'm just going to try to say what he says all the time in group meetings, which he says, you have the area of a mother compartment in the ablated scenario and the unablated scenario. They are not significantly different. You have the area in a daughter compartment in the ablated scenario and the unablated scenario. They are not significantly different. And so he says, if you want to say this protein is significantly enriched on the apical side, then he says the word really loudly in group meeting. He says significantly. And he says, if you want to use the word significantly in a sentence, if you want to use a comparative in your sentence that has scientific meaning, you must have done a p-test. And that's the rule. You can say whatever you want. You don't have to have p-tests if you're not going to use the word significantly in your sentence, or if you're not going to use a scientific comparative word in your sentence. And I think that's a really good rule, that if you want to say, I am significantly taller than you, right? Then I've got to somehow get statistics in there to justify the use of my words significantly.
Okay.
Yeah, I like that way that Eric is thinking about this. It's made me really on board with a lot of p-value and significance tests that I wasn't on board with before. Just reframing it that way. And then if you don't want to do that, don't say that sentence. Say the sentence that you meant to say. If you really want to say a sentence that says, I am, significantly faster than my friend, then you really need those to go do those statistics. Okay. Figure 5A. I can't with these figures. I just want to ask you. Figure 5A has 3 panels. They have a group of boxes on the left, a group of boxes in the middle, and a group of boxes on the right. What is the difference between the group of boxes on the left and the group of boxes in the middle, other than it's a different set of images?
Yeah, so this is where...
I just figured it out. I stared at this for, I gotta say, 5 minutes. I circled it in red and I said, same? I figured out what the difference between the 2 boxes of images are.
Okay.
We'll time you. Okay, I'm timing
there's
you.
the MRLC GFP versus Mo
God
GFP.
God damn it. I didn't notice that. They're highlighted the same color. They're almost the same letters. God damn it. All right. I like literally stared at this. I was like, this figure is not as bad as the other figures. This is like a much bigger font than the other figures. I'm like, even this figure, I can't figure out what's going on. All right, you figured it out. Yeah, maybe I'm just blind.
But I don't know what Mo GFP is labeling, so.
I looked it up. I looked up all these ones that I hadn't known. I'm trying to remember what Mo GFP labels. I think I do remember. I think Mo GFP labels something on the plasma membrane.
Okay. So this is a membrane marker.
This is a membrane marker.
Cool. Okay.
And I remembered knowing exactly what it looked up. I did a lot of research for this paper that is all...
in one ear, out the other.
Yeah. And then at this point, we're an hour in, so I'm going to read what I wrote at this point of the paper. So on figure 10, I said, at this point, I'm just exhausted. In a normal journal club, I would give up. The only reason I'm still going is because we are recording this. And then I said, we just had 2 full-page figures separating this wall of text from the header that told you what this wall of text was about. And I just felt overwhelmed that I was just, scrolling back and forth. It's from a formatting perspective, so hard to read this paper. I guess maybe what's going on here is, as you said, the online version, not the PDF version, is not that hard to read. It's still super hard to read, but it's not as hard to read as the PDF version. The PDF print version is what's hard.
Yeah.
I think I need to change my note-taking strategy so that I'm no longer going off of paper,
so
and I'm no longer going off of notes.
To read and take notes in an online, I don't know. I've never tried.
Yeah.
But
I think it's time to find out.
Maybe the Papers plugin lets you do this in some way.
I don't think the Papers plugin lets you do it. But yeah, listeners, if you have a good way to take detailed highlighting style notes on a web page that really syncs well with multiple computers and has a similar environment to Papers, let us know, because this has become untenable. On this page that I almost gave up on, I had a little jibe.
Haiku.
No. What, Linnea, is nuclear fallout?
Yes, you had hinted that this would be your jibe.
If you had to define nuclear fallout, what would you say?
I would say nuclear fallout is the aftermath of a nuclear war.
Absolutely. Absolutely. Here, they have the audacity to have a sentence. Nuclear fallout is the spontaneous elimination of nuclei from the fly blastoderm. Come on. Also, this implies that nuclear fallout is only the elimination of nuclei from the fly blastoderm. That if it was from the mouse blastoderm, you wouldn't call it nuclear fallout. Presumably, you'd call it nuclear Armageddon or something in the mouse. Scientists stop taking words that have other clearly...
it's cited by, they cite 60 to 65. I'd like to know what the earliest citation is there and whether it was during the Cold War.
Then you imagine you go to Congress and Congress is like, well, we've given you $5 million to study Drosophila, and it appears that you're writing papers on nuclear fallout. So what are you doing? Yeah, this is just always makes me think of...
But I think this is a good back pocket for a science word draft.
Yeah, I really want to do a science word draft. I mean, I guess we could do them all. But the one that I'm always thinking about is MRIs. Do you know what MRIs are?
Naked mole rats? No.
I forgot about the naked mole rats, the NMRs. Oh, that was so good. Man, there's a lot of in astronomy, when I was an undergraduate, we would study magnetorotational instabilities. And I was so upset because I was like, clearly magnetorotational instabilities were studied after MRI machines were created. What did you think? Anyways, scientists, make more words. Make new words. Don't call things nuclear fallout. If I ask a biologist if they're studying nuclear weapons and they say, yes, I study double-stranded RNA, I'm going to hit them with like Kim wipes.
Okay.
So I guess it's worth saying the end of this is super cool. At the end of this, they say, and they kept saying the word extrude, and I really couldn't figure out what the word extruded meant until the very, very end. But basically they say there are things that can go wrong with these cells that are not being captured via normal mess-ups. If for some reason the nuclear divisions and the cytoplasmic divisions go out of sync, one thing that this can cause is that the cytoplasmic division stretches the nuclei, the cell gets a kind of weird shape, and then the cell is going to pop up out of the cell layer that it's in and then get collected for destruction. I thought that was super cool. I couldn't figure out what they were talking about until figure 7, and I couldn't figure out what they were talking about until like the last page of the paper. But when I finally understood it, super cool.
Yeah, that this is like a safety mechanism of some sort, or like some sort of check of like, if you're able to keep your cell's cytoplasmic division and nuclear division at the same rate, you're doing well as a cell. And if you're not, then getting rid of you.
So coming back to what we were talking about at the very beginning, one of the reasons why I would like to know so much, can this happen in single cell organisms? Can you get clammy that don't have nuclei?
Yeah.
And then do those clammy continue to divide, or do those zombie clammy continue to divide? Is we were talking about, I made a theory about the timing of differentiation in multicellular tissues. And I'm really, really proud of this theory. I don't know if I'll ever share it with anyone, but part of the holes that you started to poke in this theory is that it doesn't handle single cell organisms very well. And my defense was like, I just don't know if it's applicable to single cell organisms, but I've been thinking about it ever since of like, is it applicable to single, and if not, why not? And one of the things that you brought up was CDK1 cycling. You were like, I don't think your theory handles CDK1 cycling correctly. And that's what I was thinking about this whole paper. Were you thinking about my theory at any time during this paper?
I was not. No, I was thinking about clammy cell division the whole paper.
Yeah. And so I guess my theory would suggest, that at the end they're like, oh, we need to search for a lot of mechanisms of what's going on, and if it's post-translational modification or something like this.
Yeah.
And my theory, I guess, would imply that their mechanisms are my bottleneck points, my thin points in the topology, and that their findings might find mechanisms, right, such as CDK1 cycling or other mechanisms, but they wouldn't ever guess with predictive power the timings of divisions. They would have to always get the timings of divisions empirically. They'd always get them after they'd find the mechanism, after they'd found the thin bridges. But that my theory would predict the timings of something like division, even with certain bridges cut, you'd be able to predict based off of the pools of the state sizes on the sides. Okay, I'm saying a bunch of stuff that means nothing to anyone who hasn't heard me rant about this for a long time, but this was something that I was really, really excited about the whole time, is like maybe these findings is a way to rescue. We just had a full pause. Everyone went silent because Linnea needed to sneeze, and we don't know how to edit these things out.
Yeah, so I mean, one of the reasons why I wanted to read this paper was so that I could do some experiment like this in clammy to try and see. So like you should be able to inhibit CDK in clammy and see what happens to division.
Do it.
And so does chloroplast division continue? Does purinoid division continue? Or is CDK upstream of all of those things?
I wonder if anyone's done it before. I suspect no, because this paper so far only has 16 citations, and it's 2 going on 3 years old now. I was quite salty that my PRX paper did not get more attention, even though my PRX paper is completely unreadable and arguably has figures that are less navigatable than this. So it serves me right. But this paper is clearly more important than my PRX paper and is getting the same amount of attention or maybe even less attention. I mean, it blows my mind that this paper only has 16 citations as of this recording.
Yeah, that's wild. I really want to do the cell cycle inhibition experiment. I should do it some.
Yeah,
maybe I'll do it over Christmas break.
I just double checked it to make sure that I'm right. On Google Scholar, cited by 16. Published 2023.
2023. Pretty shocking because this guy is a pretty big deal.
It's huge. It's good work. It's got great videos, which we'll get to put on YouTube. My complaints about it are really complaints about what cell is and not complaints about what this paper is. This is not the kind of science that I would ever do or the way that I would do it, but this is good science.
I don't know enough, and this is like part of reading this paper, and there's still a lot more reading to do, is I don't know enough about the cell cycle clocks field and what their normal models are. So is Drosophila the go-to model, or is this an unusual model paper for that? They argue that it has some unique features because it has these synchronous divisions.
It also
you could imagine that the cell cycle field, the cell cycle clock field, is like mostly in tissue culture or something.
Yeah.
And so it's a hard thing to cite.
Oh, you know what's going to be a really good model for this kind of research now? The chicken.
The chicken.
Yeah.
Okay.
Could they have this? I forget what the name of the sensor is that tells you if you're in S phase or M1 phase in the chicken. Was this new sensor?
Yeah, I mean, so that would be
Fuji. The Fuji.
Like, have people added CDK to look at developing chicken embryos?
I don't know. And yeah, my whole thing about this is like, I don't know how novel this all is. I don't know where the literature was. Yeah. It could be that the reason why this only gets so many citations is, yes, it was formatted like a Cell paper, and so they got it into Cell, but really this is all quite known before. I have no idea. And that's one of my explanations for this, is that, like, to some extent you can get papers into certain journals just by being clever with your formatting and wording. And it could be that this is not nearly as...
Or that nobody got past this first section, which you say... So, like, this first bit saying is probably pretty well known, and then nobody got to the
Yeah, the first page is super weak, and the first figure is pretty brutal. And so it's hard to make it through this. And we only really made it through this because we're being recorded. We're going on the record here. But yeah, the Fuji cell cycle biosensor that everybody at the Roslin Institute made for the chicken. And then you could go in with the chicken and mess with CDK1 and everything and find probably just the same thing that these guys found. So I guess what's the point of doing it?
Well, but conserved across organisms would be...
But they kind of know that it's conserved across organisms already. They know that it's in zebrafish, which predated this work. They know that it's in canine epithelium, which predated this work. But they know that
there's... So, yeah, okay. They know that there's these cytoplasmic divisions without nuclear divisions, and then you're assuming that it has the same.
I guess the one big thing for doing it with chicken is that you can do the chicken blastoderm unbelievably beautifully, and so maybe they can go in a similar developmental realm in a vertebrate and look at the story there. Or I don't know. Yeah, I don't know.
Yeah.
Sophie Dumont is going to do it.
I'm pretty nervous for this podcast because I feel like it's so on the corner of what we're experts in.
Yeah.
So we have huge blind spots that are going to be super apparent to all of our listeners.
Yeah.
Yeah, that makes me apprehensive.
But reading the mission again, the primary goal is for Bezier and Linné to spend time together talking about shared interests. We did that.
Yay.
Oh, I guess we actually have to do, we can't spend time reading our mission statement and saying that we should be unafraid and enthusiastic for our criticisms. We need to do our last bits of our format, which is, if you could have one thing added to this paper, what would it be?
Clammy. No.
Okay.
One thing added to the paper.
For me, it would be going in with a 2-photon microscope and getting full Z depth. And if you can do that via light sheet, sure, fine, whatever. But getting full Z depth.
Yeah, sounds good. I think most of my, I don't have a single thing to add to this paper. Most of my things that I'm interested in are following up on the paper.
In which case, clammy. Chicken. Yeah.
Chicken, is this,
that's a great place for them to be, right?
Yeah.
Is that the main thing that we're interested in is following up. So then because this is a paper that you really did pick, talking to Asya for this outro, broadly in the world, why did your mother care about this?
I cared about this for two reasons. The first is aesthetic, that cell cycle clocks are a beautiful biological puzzle, and they're something that I would like to think about more. And the second is about people, and that I often go to conferences and find it hard to talk to new people. This was a talk that I went to that I was excited about, and I was brave enough to go up to the speaker afterwards and talk to him. It's been a good experience to have spoken to him. We now correspond via email, and we saw each other at ASCB. A lot of science is connecting people, and this is a person that is doing cool science and is excited about other people doing cool science.
So then for Asya, specifically, what was this paper about? So they take Drosophila, which is a kind of fruit fly. It's the standard fruit fly in science. And during its very early embryonic development, it's just a bunch of cells that divide and divide again and divide again. And there are these beautiful movies in which it's on a very tight clock, the divisions. And they say, if you look carefully, sometimes the so-called cells that are dividing don't have a nuclei. They don't have a nucleus inside the cell. It's just a hollow zombie cell that's pretending to be a cell. And then they go in and they study how do those cells divide if they don't have a nucleus? Because a lot of cell division has to do about taking all the DNA of the nucleus, creating two copies, separating those two copies, and then dividing. And so if you don't have that whole program running, what's going on? And they basically poke at each bit that could do the division. They poke at the microtubules. They poke at the actin. And they say that this division can happen independent of this main cycler that's setting the timing on all cells. It can happen independent of the main microtubule architecture that exists in the cells, but it can't happen independently of both. And they also say that you kind of need myosin as well at the very end, which we skimmed over. And lastly, they say when this division is going on without a cell inside it, it kind of pops this cell up above all the other cells so that the cell is no longer confluent with the rest of the tissue. It's no longer part of the tissue. And so this cell was defective in some way that caused it to not have a nucleus or caused its cell cycling to go at the wrong tempo. So it had a nucleus, but the nucleus got messed up. It doesn't have necessarily DNA damage, but it has some sort of damage. And so it has to be thrown away. So that's what this paper was specifically about. They go a little bit into, the speeds, but that's specifically what the paper was about. I think that was a terrible summary. That's my summary.
I feel like it was fine. I would look at their graphical abstract. I find it helpful.
Yeah, I actually don't find their graphical abstract all that helpful, even though I've read the paper a lot.
Okay. Well, is there anything we need to communicate to our friends?
We are voting on episode 15. So the poll is back on Patreon. And if you have a better way to do polling other than Patreon, let me know. But if you want to vote for what paper we're going to cover for episode 15, you have, I think, 7 options on the Patreon. Go and vote and we'll cover that paper. We're going to go build a microscope and finally align a cylindrical lens that we've had misaligned for maybe 2 years now.
Thanks for listening. Bye.